Good afternoon, everybody. If you can get your seats, and then we can start with this session. I welcome you all to this symposium on new approaches and innovation in TB vaccine research and development. And I welcome you all on behalf of the organizers of this session, which is ERAS Foundation, which is a foundation based in Rockville in the USA, and is actually advancing the development of TB for the world, vaccines for TB. And on behalf of Tuberculosis Vaccine Initiative, which is based in Europe, having its office in Lelystad and has a footprint primarily in the European research area. And I welcome you also on behalf of my co-chair, Willem Hanekom, who's deputy head of the tuberculosis at the Bill and Melinda Gates Foundation. My name is Frank Verrek. I'm representing TBVI at this stage. And this is perhaps even a little bit of a peculiar session in the whole meeting as a whole, because it's a lot about drugs and diagnostics and practical clinical and healthcare around lung diseases and TB, but we're not hearing that much about vaccines. When we look at tuberculosis specifically, it's clear that the advances have to come from better diagnostics and new drugs, obviously, but we feel very strongly that vaccines would make a very relevant component of prevention of TB. It could really make an impact in breaking the transmission cycle and thereby preventing the spreading of the disease, and particularly in the light of the antimicrobial resistance, that it's becoming more and more a threat to also to tuberculosis disease, that vaccines can be making a difference. Now, without much further ado, I will start with introducing the first speaker of today, who is Danny Casimiro. He's the Chief Science Officer at ERAS Foundation. And he actually joined ERAS February this year. Before that, he was working with Merck and was responsible for several development plans of vaccines and biological, both in infectious disease and oncology. Now he's overseeing the preclinical work for vaccine development at ERAS, and he will tell us about the pipeline and diversifying the approaches that we take for vaccine development against TB. Please, Danny.
Thank you, Frank. Thank you very much, Frank. I'd like to thank the organizer as well for providing us the opportunity to share with you an overview of the pipeline, especially the preclinical pipeline now and in the future for TB vaccines. I hope in this talk I could impress on everyone that vaccine should be one and is one of the three pillars for controlling any infectious disease, including tuberculosis, right? Diseases like polio, smallpox, tetanus, rubella, measles are no longer a public threat because of the implementation of global vaccine campaigns. And we'll hopefully soon see more about new infectious diseases where vaccines have now entered the market. So there has been, in the field of TB vaccines, there has been a lot of progress in the last few years. And I hope to impart, to share with you the current status of the TB vaccine R&D. And we also are developing a strategy for diversifying the pipeline in order to increase the probability of success of having an effective vaccines. I'll also be discussing some new and improved tools that many in the field are working on. And hopefully by the end of the talk, I can impress on everyone that the vaccine is achievable. Right. No. Apologies. So I work for ERAS. It's a not-for-profit organization founded in 1997. It has 3 offices, one in Rockville, Cape Town, and an office in China. Our mission is to develop a new, effective, and affordable vaccines for all those who need them. We operate under a product development partnership model, and we have a lot of expertise when it comes to translational science, as well as capabilities in candidate optimization, all the way to late-stage clinical development. It's very clear that we will not meet the global targets for the epidemic without new TB drugs, diagnostics, and more importantly, vaccines, right? The priority of ERIS is to develop a vaccine to prevent TB disease in adolescents and adults. This is simply because the major source of TB transmission occurs in this population. And the key to preventing MTB infection also in the younger population can be attributed to controlling TB disease in the adolescents and adults. And in the end, it will provide a very cost-effective and sustainable solution to controlling the epidemic. So it's also key in terms of controlling AMR, right? We have to realize that vaccines are based on antigens which are fairly conserved, that are not subject to resistance due to drugs. So we all know that the correlation for development of resistance is with high antibiotic use. And if we do have a vaccine and increase the vaccination and use of the vaccine, we should, in principle, lower the use of antibiotics and the rise of antibiotic resistance. So it's really one of the benefits, and it's been shown in many other diseases like strep pneumonia in terms of the impact of vaccines in terms of reducing microbial resistance. My apologies here. So new TB vaccines are achievable. It's very clear that the human immune responses can control TB. About 90% of infected individuals develop latent form of TB or of MTB infection. There are reverters, for example, with quantiferin tests. So a number of individuals, a fraction of individuals who turn positive for QFT would revert back to being QFT negative. And there are cohorts of individuals who are highly exposed but do not turn QFT positive or resistant to TB infection as well as disease. BCG also provides partial protection. And also, in the last few years, there have been many scientific progress that would lead us to increase our confidence that we would have a TB vaccine in the future, in the near future. First of all, there are refined non-human primate models, which will now enable us to down-select candidates before they go into larger trials. We do have a promising vaccine, at least in the ERIS portfolio, in the CMV or cytomegalovirus vaccine, vector vaccine expressing TB antigens. Work is underway when it comes to game changers like controlled human infection models or challenge models for TB, as well as rigorous or robust functional assays that could substitute or inform the selection of new vaccines into the development portfolio. There are also novel clinical trial designs that will allow us to simplify, or at least to facilitate the selection of vaccines before they go into large prevention of disease studies. So I think there's a lot of progress in the last few years that lead us to believe that there are more to be done, but we are on the right track in terms of a TB vaccine. Now, the global TB pipeline is shown here. This is the clinical pipeline. There are 13 different candidates. However, they lack diversity. They all are meant or are designed to induce CD4, class 2 dependent CD4 T cells, as well as class 1 dependent CD8 T cells, the conventional T cells, right? And they are restricted to 3 types of modalities, namely the viral vector vaccines, the subunit or adjuvanted subunit vaccines, as well as the live or inactivated whole cell vaccines. Now, ERIS is working on largely the subunit vaccines at the moment, but we should note that for the target product profile that we want, which is prevention of disease in the adolescent and the adult, only one vaccine right now is in a proof of concept study or an efficacy study, and that's M72. So despite the richness, what apparent richness of this pipeline, only one vaccine is currently in a phase 2 prevention of disease in adolescents and adults. Now, why do we need to diversify the pipeline? All right. So we know at least we have a limited diversity in the current pipeline, despite the numbers that we have. But TB is a very complex pathogen. It induces several arms or stimulates several arms of the immune system, conventional as well as non-conventional T cells, namely CD1-dependent T cells, which are activated by the complex between CD1 and a number of the mycobacterial lipids and capsular proteins, capsular polysaccharides. And it also induces or stimulates mates. And there are reports that it also induces HLA-AE. So we haven't really paid enough attention to this type of the immune response or this side of the immune response that could potentially control TB. So the goal in the next few years, or one of the goals of the next few years, is to better understand the role of this type of immunity in controlling TB, and to develop approaches or vaccine approaches that could elicit those potentially protective immune responses. So these are a number of the approaches and projects that we are involved in, as well as many in the field, and we'll continue to work on in the next few years, namely novel viral vectors or vaccine vectors, aerosolized vaccines, or vaccine concepts that could induce protective antibodies, as well as non-conventional T-cell responses. We are also going to focus on new tools, right? It's important it's important to keep the eye on developing new tools, because at the moment we have fairly blunt tools in terms of down-selecting vaccine candidates. It's now that we do have non-human primate models, but those are still very expensive, very time-intensive models. So these are the areas which I'll be discussing in the next few minutes. The first one of which is CMV. Okay. Cytomegalovirus is a human virus. In this case, the vaccine is a live attenuated but persistent virus or vector. The vaccine for TB has been designed to express multiple TB antigens. It's developed by Louis Picker from OHSU, and it's also been used for the development of an HIV vaccine candidate. What's unusual about this vector is that it can stimulate conventional as well as non-conventional T cells, depending on the design of the vector. And it also stimulates tissue-specific effector memory responses. One thing that I'd like to point out is that in the case of CMV, we've seen the best protection so far using this vector in the rigorous rhesus macaque model. And the data is shown here for one of the studies that we have conducted with Louis Picker. You have a group of unvaccinated animals, a group of BCG-vaccinated animals, a group of animals or rhesus macaques that were vaccinated with a CMV-TB vaccine. This is on the rhesus CMV vector or background, simply because human CMV does not replicate in rhesus macaques. And a cohort where BCG was given as a prime, followed by the rhesus CMV-TB vaccine. On the graph on the lower half, you see longitudinal CT scan data for each of the cohort. And on the right here are the positive lung tissues for TB at necropsy, as well as non-lung samples for the different groups. It's apparent from these results that the CMV-TB provides the best protection or the better protection compared to the BCG relative to the unvaccinated macaques, both for CT scans, lung, CFU, as well as our lung positive tissues, as well as dissemination in non-lung samples. Okay, when it comes to diversifying the administration route, right? We're looking also in terms of aerosol administration. We have an ongoing collaboration with GSK as well as Transgene in terms of heterologous prime boost using vector vaccines for TB given by aerosol. And we're seeing some very high responses following the MVA booster in those animals. And we have an ongoing collaboration also with Frank in terms of testing the concept in a non-human primate challenge study. We are also interested in terms of looking at attenuated MTB strains, as well as BCG or recombinant BCG. But in principle, the approach here is, can we use these vector approaches as well as the whole cell vaccines to elicit lung-resident immunity where it may not be optimized by the conventional ID or intramuscular route. In terms of antibodies, there are evidence or pieces of evidence for the role of antibodies in controlling TB. There are studies where passive transfer of monoclonal antibodies in mice prior to TB challenge have shown reduction in TB progression or disease progression. There are more recently serological analysis of the resistor cohort, in which case there are unique serological profiles of TB-specific antibodies in those cohorts compared to latent TB. So much still has to be done in terms of understanding the role of antibodies in MTB control. Future research revolves around the role of the backbone or the effector function of antibodies and glycosylation in control of TB, especially dissemination of TB. And if there is really a strong role for antibodies, can we identify novel antigens that could be included into a future vaccine candidate? And these are coupled again by having better tools, or in this case, transmission models for evaluating this type of vaccine concepts. Slow as it is. Here we go. Frank, well in time? Okay, thank you. So this is just the last slide when it comes to the the talk itself, the body of the talk. So a game changer in TB is to develop a human challenge model, right? It's been critical in terms of selecting candidates for diseases like malaria, RSV, influenza. And unfortunately for TB, we have to rely on efficacy readout, either in large preclinical animal models, or in this case, some proof of concept or proof of biology, like a proof of prevention of infection, or worse, prevention of disease in an enriched cohort in a large clinical trial. So having the tool to challenge individuals safely, vaccinated individual, is a game changer, a quantum leap for the field if this is possible. So many of the investigators, academic investigators, are working in developing models for controlled human infection. Investigators from Albert Einstein College of Medicine, Harvard Imperial College, and Oxford, as well as Helen McShane is here. And Helen is looking into the use or development of aerosol BCG as a challenge model. A novel approach is to develop attenuated MTBs that are fairly safe, as well as fairly have the appropriate indicators for detection. In this case, in the case of this part of the consortium, there are approaches that would incorporate genetic kill switches or dependence on exogenous nutrients to limit the replication, the in vivo replication of the strain in a challenge model, human challenge model. It's important as well, the fact that you're limiting the replication, you should be able to detect those strains. So in this case, detection systems or reporter genes are being incorporated into the attenuated MTB in order to enable detection after aerosol challenge or after intradermal challenge. In the end, this will depend on whether you have a strain that is safe and sensitive and ultimately predictive of vaccine efficacy. So a new TB vaccine is the most efficient way to prevent MTB transmission and stop the epidemic. A new TB vaccine, while challenging, we believe is achievable. And why do we believe that? Because there have been considerable preclinical advances in the last 5 years. We now have a promising preclinical candidate that will move into clinical development, hopefully within the next 2 years. And we have better models now to down-select candidates. And in addition to that, we are developing and will now implement strategies to improve the probability of success by diversifying the pipeline, as well as developing new tools for selecting the candidates. So I'd like to thank the major funders and R&D partners for ERAS, our clinical sites and networks. And I'd like to thank my colleagues at ERAS who helped me prepare the slides and the presentation today. And I'd like to thank you for your attention. Thank you.
We have some time for questions. Any questions?
No.
Why do you think that the CMV vector seems to work on its own but doesn't seem to boost an effect when you give a prior BCG?
Yeah, so that's a very good question. I think we don't know entirely what the reason for that one. It could be some innate residual effect on the CMV vaccination because these are only 3 months apart between the BCG priming and the first immunization with CMV. We do plan to conduct a study where we would, in our plans, it's a study to immunize infants, rhesus macaques, and give the CMV vector at about 4 years after the initial immunization. It could also be just the original antigenic sin, right? So it's a very good question. We do plan empirically to address that particular question. So moving forward, in fact, we do feel that we want to test vaccines moving forward without BCG priming, if at all possible.
Thank you. Very nice overview. Just 2 questions. The CMV vector, it's a pathogen in humans.
Yes, yes.
So how do you see the safety issues around that being resolved? And the second question is, does any of the vaccines in the pipeline, has any vaccine shown a signal of protection in humans? And if so, what's your take on that?
Okay. I'll answer your first question. It's probably the easier one for me coming from my background here. Now, CMV, it's important to make it as safe as possible in humans. As you know, CMV causes congenital CMV disease, right? Infection and disease. That's the transfer of CMV viruses to the unborn fetus. So the vaccine or a vaccine vector has to have limited replication and, if possible, limited, if no shedding at all. If not, definitely no shedding, but no spread beyond the injection site, right? So there are ways to do that. You can make it replication incompetent, or you can make it replication deficient, right? And strategies are in place to make it at least replication deficient by removal of a tegument protein, like PP71. Yeah, very good question. This vector right here is not designed to be replication deficient, or it does not have the same mutations that are planned for the human studies. But we do plan to test an equivalent vector to the final human vaccine in a future non-human primate study. All right. There are obviously some, to your second question, could you repeat it again?
Well,
that's a very So it is, I mean, and I'll draw some experience here as well. There are definitely some reports of efficacy in humans with some of the wholesale vaccines. It's always very good to show, to understand what the correlates would be in those studies, to inform future studies. So it's always important to know that in order to, one, corroborate as well the robustness of that protection. And you've seen that in the case of HIV with RB144. No one believed there was efficacy until you did a correlate analysis, everything seemed to fall into place, right? So it's important for us, when moving forward with efficacy studies, to have biobank samples so we can go back and further understand the basis for that protection.
Would like to support a study of a vaccine that's protective in humans or shows signal if it has an associated biobank to identify correlates.
I think we can discuss this one separately. So, and obviously all of this will be dependent on our partners as well as our funders as well. Thank you.
So, for example, the MVK vaccine, I know that there's some evidence of efficacy in previous trials, and the results of a phase 3 trial will become, that's being done in China, will become available, I believe, in the next few months.
Yeah.
What would be the development steps for that vaccine? I mean, will it be available globally or outside of China? And can you speak to that?
Yeah. Probably I would not be the right person to opine on the plans of that company to make the vaccine, should it be positive in those phase 3 trial outside of China. Perhaps someone here would know more about the MVAC vaccine than I would.
So that vaccine is likely to be different. It's definitely different from the vaccine that was tested in Tanzania, which was M-Obiense to start off with. And so I don't think you can actually compare the two or think that the results from the one would be applicable to the others. I think with ERIS, there are at the moment discussions of exactly what plans there should be if there is a signal and how one would follow up. So these high-level discussions are currently ongoing about what should be done if there is a signal. But it's a very complex trial with 5 dosages of the vaccine given. 6 dosages of the vaccine to be given. So I'm not sure where it's going to go. There's another question out there.
Question regarding the CMV vectored vaccine. Do you foresee any anti-vector immunity, given that that has been a problem in the adeno-virally vectored?
Very good question. At least based on the preclinical studies, they were able, these are all seropositive, rhesus CMV seropositive macaques. So they're not going to selectively into seronegative. So at the dose that was given, it's able to elicit those type of immune responses. But there are evidence in humans that giving a live virus CMV vaccine into seropositive would require a higher dose to see a booster than if you were to use it in a seronegative or to prime a seronegative individual. So it's likely that you'll have an effect of anti-vector immunity, but it can be circumvented by a higher dose of the vaccine.
So thank you very much.
Yeah.
So our next speaker is Genevieve Enchaspé. She is, this is very exciting. We're going to look now at vaccines not only to prevent TB or infection or disease, but also to perhaps treat disease as adjunctive therapy for antimicrobials, ultimately. So Genevieve is at Transgene, a company in France, and she's head of infectious diseases there, and she has done a lot of pivotal studies in this area of host-directed therapy using vectored approaches. Genevieve, thanks.
So, good afternoon. Thank you very much. So this is my mission for this afternoon, is to try to convince you and get you more familiar with the concept of therapeutic vaccination. So, and what is this class of molecule? Could this class of molecule help in the fight against tuberculosis? So I will take you through some general concept of this so-called therapeutic vaccination, and also some example of what we have to do at the preclinical level, because very often we forget difficulty of working at preclinical level with the tuberculosis models, and with some notions of combining perhaps multiple immunotherapeutics together to improve efficacy. So this is my disclosure slide. So you've seen this slide before, and just to replace, for you to have in mind where this category of vaccine will fit. We heard about pediatric vaccine. We know it's difficult. We heard about the MVA 85A trial. This is remaining a very difficult positioning to vaccinate a young infant is a tough one, but obviously has been done in many other instances before. We've been a lot of talks going on in adult vaccination, whether you are in the latent phase of the infection, or maybe whether you are confronted to a first infection in your adulthood. And the trial that was mentioned earlier on the M72 vaccine is positioned here. And you have these therapeutic vaccines which we conceive as being used in combination with antibiotics. That could fit on different positioning. I'll get back to that a little more in detail. They could fit here in being a kind of a post-exposure vaccine. The people are already infected. You can try to vaccinate them and prevent reinfection. But we could also address this therapeutic vaccine here in the acute phase of the disease. So on the next slide, just some general concept. If you look in the literature, therapeutic vaccines is manipulation of the host, of the immune system of the host in an antigen-specific fashion. And this is in contrast with manipulating the immune system in a non-antigen specific fashion. So there are two ways to do that, and I'll show you in the next couple of slides what is the field, where is the field standing. You can try to do it in a positive way to try to reprime the immune system, to boost the immune system in a positive way to fight against cancer, chronic infectious disease. There are many examples of work going on here, but also less known perhaps is to manipulate it in a negative way and to use this therapeutic vaccine to control, for example, autoimmune disease. There are also some efforts there. So this is shown I'll show you in the next slide. So if you look at what really is the aim of this therapeutic vaccine, of this class of compounds, what we want to do is to add a different mechanism of action that what is being used by the small molecules, whether they are direct antiviral molecules or whether they are antibiotics in the case of TB. We would like to enroll the host immune system to complete the work, maybe, that is difficult to be done with some of the antivirals or antibiotics. And obviously, we are going to apply this vaccine in the face of an ongoing infection, whether it's active or latent. That's a major difference with a prophylactic vaccine. I'm not saying a therapeutic vaccine have to be different from prophylactic vaccines, but the aim is different. This is novel immunotherapeutic. So coming back to the immune correlate of protection, you have to know, you should have some idea at least to what kind of immune response you want to induce or how you want to manipulate this response. And here is the same challenge than for the prophylactic vaccine. You need to have some idea of your immune correlates of protection or resolution. And obviously of key importance in this positioning, in contrast with the prophylactic vaccine, is you do not want to exacerbate inflammation and disease when you apply this vaccine. So in this next slides, I show you some of the efforts going on in this field in a general way. I mentioned that infectious disease obviously has been the target of this vaccination. There are many efforts going on. Maybe you have followed some of that in hepatitis C. Hepatitis B is a big field right now. Chronic hepatitis B, chronic hepatitis C. the herpes virus family, and obviously Mycobacterium tuberculosis fit in here, and you see that not that many antibacterial vaccine here in that range. Cancer, autoimmune disease. So I think at that age, everybody, and particularly the cancer people, are ahead of, I think, the field. We know that there's going to be a need for combination of these vaccines with the current standard of care. In cancer right now, we are combining with chemotherapy, we are combining with the checkpoint inhibitors with very interesting data at the preclinical level, and those are being brought in the clinic. So obviously in the TB field, we need to have an open mind and we need to think about combining this vaccine. This is a kind of technology that we are using. On the right part here, you see some of the technologies can be difficult and not easy to apply in a broad range in mass vaccination, whole cell tumor vaccine, dendritic cell vaccines, anti-idiotype vaccines. But some of these other technology are probably more familiar to everyone. These are very kind of standard technology that we are using in the development of prophylactic vaccines. You heard just before the CMV vector, there are a number of viral vectors that are being used. Poxviruses, adenovirus, CMV, lentiviral vectors are also being used right now. One of those vaccines is in the market in China with an adeno P53 in the head and neck cancer here, to mention that. DNA vaccines, and a lot of recombinant adjuvanted vaccines, classical ones. And I'd like to mention or bring to your attention the recent success from the GSK company, which has some similarities to TB, not too many similarities, but this vaccine here was administered to the elderly population and had a very strong efficacy, over 95%, I believe, in controlling resurgence of latent VZV. So you could really completely abrogate in the older population by vaccinating this population carrying latent varicella zoster vaccine reappearance and reactivation of VZV. So this can be achieved and has been very nicely done recently for this indication. So on the next slide, some issue. I don't know where I turned to. Okay, I just wanted to mention that. Never mind. It was just a two technology that we see in the TB field right now. Here they are. We are working on this field with the viral vectors and this kind of technology. Next slide. Oops, I'm running into the same problem you had. This is a schematic representation taken from a review by Sean et al., which is showing in a schematic way the different checkpoints in the immunology or immunity against tuberculosis. The early phase with the importance of innate immunity, I think it's recognized in the early adaptation of the host response to the infection. We know that the adaptive immunity, in particular the T-cell immunity, is very important in controlling latent TB. There has been a few antigens identified. If you develop a T-cell response from these antigens, you can control the TB. So this is when there is a break in this adaptive immunity linked to a number of issues, co-infection with HIV, immunodepression, and so on, that you lose the control of the infection, and you see this reactivation. There's a number of papers that have shown that you have loss of the T-cell response, you lose a cytolytic function, and obviously we also know in TB that there are many repressive or immunodownregulator pathways. Tregs, PD-1, IL-10 are very important. The question of antibodies is still open, but those are all the negative signals associated with resurgence of infection. So if you look at therapeutic vaccine, this is where we think this vaccine can have an impact during the chemotherapy. While chemotherapy can control, in the best cases, can cure TB infection, even in drug-sensitive and obviously in drug-resistant infected patients, we know that this cure here can maybe not definite. Oops, this is not obvious to play with this. I apologize. Okay, we know that this cure can lead to rebound or reinfection, or they either sustain or can lead to rebound or reinfection. So you will see in a couple of next slides that the therapeutic vaccine are aiming at trying to reprime this immune system, perhaps boosting it, but repriming it in an efficient way so that in combination with chemotherapy or sometimes positioned after the chemotherapy, at the end of the chemotherapy, we'll try to prevent this rebound reinfection and maybe shift that curve here towards something going faster. So these are some of the general concept. I don't know if this is really... Okay, this is some of the general concept in the field. I want to illustrate what we've been doing in the company, what other actors in the field have been doing, how do we approach developing therapeutic vaccines. So we've been working with MVA. We know MVA is a very safe vector. It's been used for vaccination against smallpox. It has a very high plasticity. You can really include a lot of antigens in this vaccine. We've included a number of antigens from the different phases. We know there is a gradient of expression, but in fact, in any phase of the TB infection, you have expression of the active, latent, or resuscitation. So we've developed a number of candidates here shown on the next slides. We've worked with more than 20, 25 different MVAs. We ended up selecting 6 of those, which have the characteristic of being genetically stable, which is very important. If you move into manufacturing, you have to have genetic vectors, and that's a big issue when we start putting a lot of antigens. You see some of the recombinant MVA carry 10 different antigens expressed under various promoters. We've done a lot of work showing that they are immunogenic in naive mice. We've looked at T cells and different measures. I won't go through that today for sake of time. I just want to show you how we've brought this into some preclinical model of therapeutic efficacy. So this is a slide that has been shown by other group and used by other group. And here, this is work has been done in collaboration with Dr. Nuremberger at the Johns Hopkins University, who is very well known and has used these models and variation of these models to look for new drugs. We've worked with them in running some of the efficacy experiments. So the principle of these experiments is to use mice, to infect mice with with a TB strains, and at the peak of infection, when the infection is established, you administer a suboptimal regimen of antibiotics. A group at Johns Hopkins has worked on many antibiotic regimens. They know how to calibrate that, and they know how to calibrate the regimen so that after about 10 or 12 weeks of treatment, you basically have eliminated most of the bacterial, become basically undetectable, but yet when you stop the treatment, you have a rebound in the majority of these mice. So this is a mimicking of rebound that you would see after antibiotic treatment, if you wish. What you are hoping to do by modifying this treatment, for example, by adding a therapeutic vaccine to it, is to bring down the level of rebound and to bring down the level of CFU and inflammation in the lungs. So this is as a proof of concept. This is a model. It's not perfect, but you like to see whether you can achieve some kind of efficacy or early efficacy. So to show you more specifically the kind of design, I just want to attract your attention on the fact that these protocols are very long, 24 weeks or 30 weeks sometimes, if you want to look at really leaving the time for the rebound and look at these reactivations. So in our particular case, we have administered the vaccine here 3 times during the treatment with antibiotics, and after the stop of treatment, different measures were performed, in particular CFU counts in the lungs and in the spleens. I'll just show you one piece of data for sake of time, and I'll come back to some data that others have published here. So this is to show you the result with the 6 vaccines that were brought in this model. This is the results of the mice receiving the antibiotic treatment alone. In the rebound mouse, you have a high load of bacterial here. This is the vector alone, and these are the different gradients of vector. What we see here is that not all vaccines are equal, and that 3 of those vaccines gave statistically significant difference in terms of viral bacterial load in the lungs. This is another way to show this here. It's just the other way around to see the protection here in logs. So in particular, that vaccine here is interesting because you see that some of the mice have really nearly reached the negativity. So out of those 6 vaccines, there is one vaccine that is of interest here that is showing this early efficacy. Obviously, we need to work more on this, but again, keep in mind that on one hand, we have 12 weeks of antibiotic treatments. On the other hand, you give the vaccine only 3 times, perhaps yet not the best route of administration of the dose, and you can already start seeing an impact on this bacterial load. Other people, very well-known groups in the field, I have worked with this kind of approach. Oops. Okay, I want to cite 3 of them on these slides. The vaccine from the Serum Statum Institute, the H56 vaccine, which is adjuvanted, is containing a protein of 3 antigens of the TB. The VPM vaccines, which is a modified BCG, and the vaccine from the IDRI group in Seattle. Those are vaccines that have actually been developed mainly and primarily to go into prophylaxis, and they are currently being tested in clinical trial as prophylactic vaccines, but they have also been repositioned as potential therapeutic vaccine. This is just to show you the kind of experiment that and the data obtained by this group published in the similar type of mouse model I've shown you before, the comparison of the viral bacterial load in the lungs after the rebound of the infection here. And you see that on average in this model, there is between 1 log to 0.5 to 1 log difference. You can compare this experiment. They have not been run at the same time, but there is in all cases some effect which may appear small and minor yet, and which may indicate that we have not yet identified the optimal either vaccine design or yet the vaccine administration. And we are right now looking at potential routes of immunization that may have an important impact on this efficacy. But interestingly, and this will be to conclude the talk with a couple more slides, I come back to this. So right now, if I'm correct, if I'm updated, which I may not completely be, but those two vaccines are in clinical trial, or they will start clinical trial. The VPN vaccine, I think the trial has started or will start pretty soon. In India, it's positioned after the end of chemotherapy in drug-sensitive patients, looking at prevention of rebound or reinfection. I think there are a few thousand patients in this trial, which is starting, which is just about to start or has started. While the H56 vaccine is a slightly different positioning, will be positioned in combination with the chemotherapy in drug-sensitive patients at first. And the final plan for this vaccine, as shown in the NIH vaccine trial site at least, is to go into drug-resistant patients to look at influence on the kinetics of chemotherapy and also the rebound afterwards. Interestingly, I think that, so this is one application of these so-called active immunotherapeutic vaccines. But as you know, and I want to conclude on these two slides, there are other immunotherapeutic approaches brought in the field of tuberculosis. This is what we have been called in the literature as host-targeted therapy. A number of molecules have been used for a number of years to try and at least to manipulate the immune system in a non-specific way, to try to ameliorate the severity of disease or to potentially cure. Cytokines, antibodies, vitamins have been used in some trials with more or less some success, not always very probing, but there is some progress and some effort being carried in that field. And it's interesting to think, there's recently a review published in The Lancet, that in fact, if you look at the different classes of molecules, they are very different. Some of them are repositioning from other fields, for example, the inhibitors of myeloid dendritic cells, or the cyclophosphamide inhibitor. I've told you that there are a number of inhibitory pathways in TB. You can try to target those with these non-specific molecules. And in fact, the vaccine with the H56 I mentioned before is combined with a COX-2 inhibitor. So you're now combining an immune-specific and non-specific vaccine, perhaps to enhance the capacity of both types of molecule. In this review, you have here the vaccine, the therapeutic vaccine, and the question would be certainly very interesting to tackle at the preclinical level whether you can improve efficacy of those vaccines if you combine them with HDT. So I think I'd like to conclude this presentation by a few statements in the conclusion, if I can move to that. I think, and we are a number of us thinking, that these therapeutic vaccines, as in other indications like cancer and chronic hepatitis B right now, have really a potential to be combined with standard of cares. And there are some early evidence, and the bulk of early evidence in preclinical model is increasing. We soon will have some data from the clinical trials I mentioned. It is not, it's one point that has been seen so far. I have not seen any report on the capacity of the potential deleterious effect of this vaccine in terms of exacerbating the inflammation. That hasn't been an issue for the moment. We know that T cells are implicated. We don't know the fine mechanism of action. This is in study today. And I think that it will be very interesting indeed to see whether what we will see in the clinic, thinking that in fact there was an earlier session before talking about a universal vaccine, and perhaps preventive vaccines or therapeutic vaccine can have an effect both in prevention and in a therapeutic positioning. So this is a very interesting thing, I think, a field for development of future combinations. So thank you very much for your attention. I'd like to thank everybody who has collaborated, in particular our collaborator at Johns Hopkins for the transgene project, who have been able to carry lots of experiments with a lot of mice. These trials are very long and they take a lot of time and very difficult to measure. So thank you for your attention.
Thank you very much, Geneviève. Make time for one or two questions there, please.
That's a very interesting model. Sorry, that's a really interesting model, and in a sense, it seemed like as if you were targeting patients who had completed therapy who were kind of likely to fail, and that's a hard group to define. But it really does, there was a recent study that came out of Gerhard Walzer's group that showed that there appeared to be, at least by RNA signature, viable TB in the sputum of patients. Would that be a way to sort of flag those people who might be likely to fail? That is, it would seem like one of the targets for such a vaccine would be people who completed therapy, we would be worried might recrudesce.
Yeah, absolutely. Yeah, I mean, ideally, if you could identify a signature of people who are very likely to fail, certainly I think that would be very advantageous to the positioning of such vaccines. So yes, you need to work side by side the biomarker signature, which is still a very, very complicated and open field. But yes, in fact, there are groups in the TBVI and TBVAC. There are consortium who are trying to talk to each other more and more in, discussing what are the signature and how can you benefit from this signature in the vaccine development and probably in the drug development too. Yes.
One question over there.
Hi, Geneviève. You mentioned route. And in your experiment that you showed, you gave the vaccine systemically, but have you looked at mucosal or aerosol or intranasal immunization?
Yeah, we have done one experiment, but not with the right vaccine candidate. So because it takes so long to run this experiment, you take some risk. Sometimes you start another experiment without completely waiting, you bet. So I cannot conclude on that, but yes, that's certainly something that should be done, I think, and we may improve very much what we see by a priming subcutaneous and boosting by the IN may give us a completely different picture. Yeah.
Thank you again, Geneviève.
Yeah, thank you very much.
For the sake of time, we have to move on.
Thank you.
Thank you. And the next speaker is Helen McShane from Oxford University. Helen, apart from being a well-respected colleague in our network at TBVI, she is a deputy head of the Nuffield Department of Medicine, professor of vaccinology and Wellcome Senior Clinical Research Fellow, as well as an honorary consultant physician in HIV medicine. And I think Helen will take us to the port of entry now and try and see what efforts we can do to imprint the immune system towards the pulmonary mucosa. Helen, please.
Frank, thanks, and thank you for asking me to speak today. So I'm going to describe some of the work we've been doing in Oxford, looking at delivering TB vaccines direct to the mucosal surface in the lungs as potentially a way to improve the efficacy of candidate vaccines. That's my declaration. Okay, so it makes intuitive sense that delivering a vaccine to the route of entry of the pathogen might be the best way to protect. And there are there's considerable animal evidence to suggest that this route is more effective than giving a vaccine systemically. There are lots of drugs, particularly for respiratory medicine, that we already deliver into the airways. And there are a number of logistical advantages to delivering a vaccine by inhaler rather than by a needle. I think it's important to point out this is not a new idea. It's difficult to have new ideas in TB when someone's done it 100 years beforehand. And there's a wonderful paper in the literature where Rosenthal et al. In 1968 nebulized BCG to a room full of medical students. I'm not sure this was with fully informed consent, and looked at their skin test conversion. And in fact, most of those medical students did skin test convert and developed a positive skin test. So we thought we'd apply a little bit of GCP to this. Oh, okay, I'm right. We thought we'd apply a little bit of, I didn't touch anything, GCP to this, and used this aerosol nebulization device. Maybe I'll come away from that. To deliver firstly MVA85A and then most recently BCG by nebulizer to the lungs of healthy human volunteers in Oxford. So the first trials that we've done have used this vector, which many of you are very familiar with. Genevieve has just nicely introduced MVA, and this is one of the first candidate vaccines we developed in my lab in Oxford, MVA expressing antigen 85A. So the trial design was we took 22 BCG vaccinated adults and they were randomized to receive either a low dose for safety reasons of MVA-85A, either by inhaled route using that nebulizer or intradermal administration. And all the participants in the study had a bronchoscopy 7 days after vaccination so that we could look at mucosal as well as systemic immunity. The safety profile or the safety measures are listed there. We did detailed pulmonary function tests in this study, and the safety profile was excellent. So what we found when we looked in the bronchoalveolar lavage fluid, and here we're looking at antigen-specific CD4 T cells to 4 cytokines known to be important, Th1 cytokines and Th17 cytokines, known to be important in protection against TB. And perhaps unsurprisingly, we found that when you deliver a vaccine direct into the lungs, and that's the A group here, the cytokine response to antigen-specific T cells is significantly higher than if you deliver a vaccine intradermally into the lungs. That's probably not surprising. But what was important was that we found when we looked in the blood, the immune responses after aerosol administration were at least as high, if not trending towards higher, than the group that was received the vaccine intradermally. And this is important because there's no sense in which we are compromising the induction of systemic immunity when we deliver a vaccine to the lung with the aim to induce potent mucosal immunity as well. So one of the other things we observed in this trial, and this is one of the reasons really to do this kind of experimental medicine study, was that when we looked at antibodies to the vector, and we've already had a question about anti-vector immunity and how that can negatively impact on particularly the use of viral vectors as candidate vaccines. When you give this vaccine by aerosol, you don't seem to induce systemically in the blood any anti-vector antibodies. In contrast, when you give the vaccine intradermally, you can see a very clear induction of antibodies. So this sort of got us thinking and made us wonder whether you could do what we've called homologous boosting by heterologous routes. So can you overcome anti-vector immunity by tricking the immune system and using the different routes of immunization? And we thought we'd test this functionally in this following study. So we had 3 groups of subjects. The first group received MVA by aerosol and then were boosted systemically. The second group was the other way around. And then the third negative control group, if you like, received 2 intradermal immunizations. We know that all these immunizations were given 1 month apart. And we know that giving 2 MVAs 1 month apart does not boost. So you've got significant induction of anti-vector immunity that inhibits that second boost. So this data is still unpublished and we're still analysing some of the anti-vector data, but essentially the bottom line and the punchline is that we did not see boosting. So you can see here in the heterologous routes arms, you can see that we really didn't see any significant boosting of the insert response. And in fact, that is at least in part because we saw the very clear induction of anti-vector T cell responses in the blood. So this anti-vector immunity that is inhibiting your boost is likely, at least in part, to be cell-mediated rather than humoral immunity. So one of the questions I'm often asked is aerosol immunization safe in latently infected people? And this is an important question to address before we take this route of immunization to further studies to test in high-burden countries. So we're currently doing a study where we're looking at exactly that. So we have 15 subjects in this trial. The first 3 will receive a low-dose aerosolized MVA, and then 12 subjects will be randomized to either get aerosol or intramuscular MVA. We're doing bronchoscopy. We're also doing CT scans and full pulmonary function tests to really ensure that we don't miss any safety signal in this study. I think it's fair to say enrollment in this study is pretty challenging, but we have enrolled 1 subject to date and they're completely fine. Okay, so to move on to the work we're now doing, and Danny mentioned controlled human infection models. The work and the study we're now doing is looking at, can we deliver BCG by aerosol into the lungs of healthy human people for three real reasons, actually. So firstly, giving BCG by aerosol may be a more effective route of delivery of BCG compared with intradermal immunization, and there is some non-human primate data from Sally Sharp's lab to show that that may be the case. Secondly, is an aerosol BCG delivery a useful human challenge model by which we can test or down-select which vaccines should progress to field efficacy studies? And that's really the main reason that we're doing the current study, is to try and evaluate that as a potential human challenge model. There's a third reason I'm interested in giving BCG by aerosol into the airways. Is this a model of human pathogenesis which we can use to do detailed immunophenotyping studies to really define immune mechanisms of protection in this sort of human experimental medicine setting that would support the data we have from animal studies? Okay, so this is the study we're currently doing. So it's a clinical challenge study to evaluate the controlled human infection with BCG administered by the aerosol inhaled route. And we're comparing both the safety and the immunogenicity of this with intradermal BCG immunization in healthy BCG-naive UK adult subjects. So the trial design, so for safety reasons, we've started with a very low dose, 10 to the 3 CFU by the inhaled route. We then progress, dose escalate to 10 to the 4, and then we have a randomization arm where we have 24 subjects randomized to either get 10 to the 5 BCG by inhaler or 10 to the 5 BCG by intradermal administration. Because this is a safety study, we're using the MHRA first-in-man rules, so we're vaccinating the first volunteer. We then have a minimum 7 days to do a safety review before we enroll the rest of the group, and then ditto for Group B, which was that 10 to the 4 dose, and then we proceed to Group C. So the point we've got to in this study is we fully enrolled in Group A and in Group B, and we have enrolled 7 subjects randomized to either Group C or D. So the outcome measures in the study, firstly, primarily is safety. The second outcome measure, and this is because the main aim of this study is to look at the utility of aerosol BCG as a human challenge model, is the amount of BCG we can recover from the bowel fluid. So all of the volunteers in the study have a bronchoscopy 2 weeks after infection. And then we're also looking at immunogenicity because of that sort of parallel aim, which is this a more potent route of immunization? So the status of this study, as I said, we've screened 16 and we've enrolled 13 subjects. The bronchoscopies are all complete on all those enrolled subjects. We've got no macroscopic abnormalities. All of our subjects have completed to day 28 with no serious adverse events. And that was all going just fine until we ran out of BCG. So as everyone in this room, I suspect, will be very familiar with, it doesn't seem possible to get hold of any BCG, either for clinical use or for experimental use like this. At all at the moment. Okay, so the safety data to date in this study is excellent. So here we have solicited adverse events. These are from diary cards. Group A is the low dose, Group B is the intermediate dose, and then C and D are the 10 to the 5 dose. This is the aerosol dose, and this is the intradermal dose. All the side effects in this intradermal dose are because one volunteer had a intercurrent upper respiratory tract infection. So the safety profile is really excellent and is very comparable with intradermal administration. Certainly, we've seen no safety signal that are concerning so far. Okay. So this is looking at unsolicited adverse events, and again, you can see there's a really very acceptable safety profile. And we've also done full respiratory function tests in this study and have seen no changes in either baseline or normal predicted for any of the subjects to date. So after a lot of work and discussion with the regulators and with Public Health England, the UK is now importing BCG Bulgaria from Intervac. So we now plan to finish this study with a different strain of BCG. Now, of course, what that means is we have to go back a step and bridge on a slightly lower dose. So the design of the revised study is that we will have We will have 3 subjects who receive 10 to the 4 of the new strain of BCG, really just to bridge on safety. And then what I'd like to do now is dose escalate higher than we had before, because we have a very acceptable safety profile and because I suspect we're nowhere near the maximum tolerated dose yet. So we're going to then enroll 3 subjects with 10 to the 5. Assuming that safety profile is fine, we will then enroll 3 subjects with 10 to the 6. We will look at the safety profile in this arm and this arm, and we will then select the best dose to give then the randomization arm, where we have 12 subjects, 9 from that previous slide, and then, 3 from the previous slide, and then another 9, to either get aerosol 10 to the 5 or 10 to the 6, and then the corresponding intradermal arm to then complete that comparison. So this study is ongoing. There's another option for this study then, which once we've completed this, which is then to add a BCG vaccinated arm to this study. This would then allow us to compare the recovery of BCG from the airways in a group of subjects who previously had BCG compared with a group of subjects who haven't. And the aim of that would be to see if we can use that model to detect a BCG vaccine effect, because that would give us some biological validity that this controlled human infection model is actually measuring something that is biologically meaningful. So to pull all that together, I think our data to date suggests that aerosol vaccination is well tolerated. We've seen with the studies with MVA-85A that aerosol delivery is a potent route of delivery, both to induce mucosal and systemic cell-mediated immune responses. And I think we clearly need to do more work on both BCG and heterologous prime boost vaccination, and of course studies in TB endemic countries to see whether this early promise from these early studies is realized. I just need to thank all the people who've done the work, my group in Oxford, both past and present, my respiratory medicine collaborator, Henry, who does all the bronchoscopies for us, and the various funders of the studies I've presented. Thank you.
Are there any questions?
If you use the oral route, do you get a similar response?
Yeah, it's a good question. We haven't done any studies looking at that. The only data on that really is Dan Hoft, who many years ago did a study where he looked at oral BCG. And from what I recollect, he did see immune responses, but he had to give very high doses because there's a great deal of loss and obviously breakdown in the stomach. Yes. I mean, I think the aim of the aerosol work is really to focus it on the respiratory mucosa, because that is the route of entry of the pathogen in humans.
Helen, have you managed to look at things like tissue residence,
absolutely.
immunity, or
Absolutely.
you're not there yet?
So we are somewhat constrained in what we can do in people compared with mice, but we're very keen to look at that. And in fact, in the amendment which we're going to put into this study for the BCG study to change the strain of BCG, we are going to also put in that we will do endobronchial biopsies, which my respiratory medicine collaborators tell me is perfectly acceptable, are done in all the asthma studies, have an acceptably low morbidity rate in healthy volunteers. So I hope that we will be able to use those lung biopsies to then compare bowel responses with lung responses, which is, of course, what you can do in the mouse, but can't yet do in humans. I mean, I think another way to address that is to look in non-human primates. And I think that's another reason why actually parallel human and non-human primate studies can really, because then you could bridge on bowel and blood, could really give us more information in this space.
Excuse me if my question sounds too naive, but I'm just curious to know, why can't we have a prime boost both by the aerosol route?
I don't think I said that we couldn't. We're, in a sense, these are primarily safety studies to start with and proof of concept studies. I think given that we see no diminution of the systemic response when you immunize by aerosol, then in theory, at least, there is no reason why you couldn't give a prime and a boost by aerosol route, I agree. But we haven't done those studies yet.
So thank you very much, Helen. So our final speaker today is Tom Scriba. He's from SATVI, the South African TB Vaccine Initiative, where he leads the the immunology side of that side of the effort. He's been doing translational immunology studies and obviously a leader in this field in the world. So we're looking forward to hearing from you about what protects us against tuberculosis.
Thank you very much. I wish I could speak to you about correlates of protection. Unfortunately, that would require a vaccine that actually protects and that we can use in trials that are placebo-controlled to demonstrate what part of the immune response is responsible for that protection. So I have to settle and talk to you about correlates of risk. I hope I can drive this thing. It seems like it's going to be difficult. Let's see. So I think we all accept now that there is this major spectrum of TB and that it's a very complicated set of outcomes where, of course, if we become infected, we may actually resolve that infection spontaneously. At least that's what we believe may be possible. We may also contain the infection at lower levels such that we can't detect the bug, but we can only detect the immune response to the bug. And then we can possibly see subclinical disease using either PET-CT scanning and perhaps some biomarkers now. And of course, active TB disease, we are trying to feverishly diagnose so that we can treat people. And so we can use the spectrum in the absence of a protective vaccine. Of course, we can try and use the spectrum to glean what is it about particular characteristics of the immune response that may help people move perhaps in this direction of this pathway instead of that direction. And so the first question that I wanted to address to some degree is whether antigen-specific T cells can protect against TB, or at least can prevent people from moving in that direction. And so it's amazing that many of the people that contributed to this work are sitting in the audience, so I hope I'm going to do a decent job at conferring the information. The first one was done a number of years ago, led by Willem, actually, which was a correlative risk study after BCG vaccination in infants, where a large number of infants who received BCG at birth, as is routine in South Africa, we collected blood from them at 10 weeks of age and then followed those up and identified babies that developed TB during 2 years of follow-up or not. And so we could do a progressor, non-progressor comparison to look at the immune response in those. And so the take-home message from this study, I'm summarizing just in one slide, is that we found absolutely no difference in the frequency or the functional profile, cytokine co-expression profile of T cells in those who went on to progress compared to those who did not progress. So essentially, we could not see any kind of correlate of a progression in this study. Yet, when you read the many reviews that are out there about immunity then against TB, then many people say that, that Th1 cells are critical for protection against TB. And I think this comes largely from studies where humans, for example, have deficient Th1 responses because they may have mutations in the IL-12 or interferon gamma pathway, or from animal studies where we can knock those out. And so, of course, these are pretty severe disruptions of these pathways, and perhaps the more subtle frequency differences or functional differences are just not that important. However, in the wake of the MVA-85A trial that we performed in infants, which of course did not protect over and above that protection that is afforded by BCG, we collected valuable samples from infants that developed TB disease in that study. So there were 55 progressors, and then we could match a large number of controls to those progressors. And again, we could look at pre-vaccination time points. So even before they got MVA, of course, they received BCG at birth, to also, again, look at can we find differences in the immune response in these groups that tell us whether there's a difference and whether some of these immune responses may matter. And so Helen Fletcher and Helen McShane and many others in the room were involved and led these analyses. And so what was really very striking was that simply by looking at the frequency of T cells that express interferon gamma in the blood when they are stimulated with BCG, the people that developed TB disease, the infants that developed TB disease had lower frequencies of these cells compared to infants that did not develop TB disease. So perhaps not that surprising for some in the audience. It looked like these T cells, these Th1 cells, are actually perhaps contributing to some kind of effect that prevents progression to TB. And in the same study, we recently had the opportunity to investigate levels of interferon gamma expression, this time not to BCG, but in the quantiferon assay to ESAT6 and CFP10 stimulation. So, of course, we did these assays to control for infection in the trial. And the vaccine had no effect on infection, as far as we could tell. But what was really interesting, and this was done in collaboration with Jason Andrews, who's at Stanford University, when we looked at the levels of quantiferon, interferon gamma expression, there were some very high levels in some of the babies. And the red bars at the top here that are very inconspicuous actually depict babies that develop TB disease. These are, this is the measurement that was made at 1 year after vaccination. So when we stratified babies at 1 year, according to whether they developed TB or not after that, so this is a predictive marker, we observed that babies that had really high levels of interferon gamma in response to ESAT6 and CFP10 were at much higher risk of developing TB. Whereas the babies that had lower levels, and these are the blue bar or the blue line indicates babies that are quantiferon positive but has a low level of interferon gamma production. So here the quantitative level of interferon gamma in this assay was an indicator of risk. Okay, so the take-home message from this first part is that it does look like the magnitude of antigen-specific T-cell responses can, in some cases, predict risk or risk of progression to TB. Of course, the antigenic target is critical. In the first instance, it was BCG, and in the second instance, it was ESAT6 and CFP10, which of course indicates infection. So I think it's safe to say that we really need to go much further. We need to look at different functions, and of course people are doing that, but I think it's still quite limited, what we are doing as a field in terms of addressing these concepts. The phenotypic or the phenotype of the cells, the location. Helen's talk was excellent, and we really need to look more at the location of these cells. And then, of course, there are many, many other components of the immune system. And in fact, in the correlate analysis of the MVA trial, many, many different markers were looked at. And many of them did not turn out to be predictors of risk. But the one, of course, that was quite surprising and very interesting is that the level of antibodies, IgG antibodies, that recognize the antigen in MVA, antigen 85A, were again lower in those babies who went on to develop TB. This is really interesting because it was even observed before MVA was given, so presumably induced by BCG, perhaps acquired from the mother. We're not quite sure, we can't tell. But again, this is suggestive that perhaps antibodies, antigen-specific antibodies, may actually play a role in protecting against TB, something that I think many people are very skeptical about, and so much more work is definitely needed. Another, I think, very important and interesting finding was that the degree of T-cell activation, so here we're looking at HLA-DR expressing CD4 T-cells, was correlated with risk. So babies who had higher levels of CD4 T-cell activation were at higher risk of developing TB than babies who had lower levels of, and so that you can see here, there's the odds ratio, which was quite significant. And so this was really interesting, and so the mechanism of this isn't completely clear, but there are suggestions that it may be concurrent viral infections that are associated with this. So we wanted to go further and see whether we could reproduce this in another study, and some of you may have heard about the adolescent cohort study, where again, this is a natural progression study. Here we studied 6,300 odd adolescents. Many of those were infected, roughly half. We followed them up for 2 years, and during that time identified progressors and controls. And then we could go back to the early samples from these progressors and look longitudinally what happens during progression as the one group develops TB while the other one controls their latent infection. And so the interesting thing was that we could again see that activated T cells depicted by HLA-DR expressing CD4 T cells were elevated in those who progressed to TB compared to the controls. Now, in this case, the mechanism may be quite different because, of course, these are infected adolescents, whereas the babies, as far as we could tell, were not yet infected with TB. So the mechanism may be different, but it's quite interesting that in both studies, we saw that T cell activation was associated with greater risk of TB disease. Of course, the other thing we did is we did transcriptomics. We used omics approaches to really delve deep and ask, well, without knowing what we should look for, perhaps we should try and discover new things and get the data to tell us the new hypotheses that we should be exploring. And so I'm just showing here that many genes were either increased or expressed at higher levels or lower levels, higher in red, lower in blue. And what we're looking at here is a timeline as people progress from latent infection to TB disease as you go from left to right, and you can see that there was a large number of genes that were significantly expressed in progressors compared to non-progressors. So the way we went about this was to say, well, there must be some kind of triggering event that basically moves a person from the latent infection to the progression state. And so we looked for gene expression that indicates this kind of event that then makes people progress. But we actually couldn't find any of those kind of signals. And perhaps the sample size wasn't big enough, but also perhaps these are just not prominent signatures that we can find. In fact, what we saw much more are these sort of progressive increases or decreases in gene expression that suggest that perhaps a person is moving through this timeline of TB that I showed in my second slide, where it just progressively becomes more like TB disease. And so I wanted to show you a few examples of that, and just so that you understand the kind of figures that I'm going to illustrate. What we're looking at here, each dot represents a progressor sample that is matched to two controls. And so essentially what we're looking at is a log2 fold change between progressors and non-progressors. And you can see that these dots start ending up on the higher side as we move here from many, basically 2 years before TB, to the time of TB diagnosis where these individuals had microbiologically confirmed TB. And so what we can do is fit a line to that so we can see what happens over time, and then we can also estimate a confidence interval, which doesn't show up very well on this slide, but I think my next slides will look a little bit more better. So here I'm showing just 4 examples of different types of genes that change as people progress from infection to TB disease. Here we have STAT1. I think many of you know that STAT1 is upregulated by interferons, type 1 and type 2 interferons. And we can see that this actually happens really early on. In fact, if we use the 99% confidence interval over here as a measure of statistical deviation from 0, then this would suggest that 500 days before we can diagnose TB in the progressors, we already see elevated expression of STAT1. For GBP1, another protein or gene that's upregulated by interferon signaling, we see a similar thing. Very, very early, these things are happening. If we look at more generalized inflammatory genes, so here we have ICAM1 or MyD88. MyD88 is one of the big transcription factors that's downstream of, or adapter molecules that's downstream of TLR signaling as well as IL-1 signaling. You can see that happens much later, maybe within 200 days of diagnosis of TB, and it's the same thing here. ICAM1 is a monocyte-associated gene. So what we can glean from this is that perhaps some things happen really early and other things happen later. And so if we try to summarize what's really going on here, and here we're using modular analysis where we basically look at many genes that act together, we can then really, with much greater statistical power, say what's happening as people progress to TB. And you can see that the inflammation module, so this is myeloid inflammation as monocytes or or those kind of genes become inflamed, this happens really much later than the interferon module, which is really upregulated, according to this analysis, up to 2 years before we can diagnose TB. So it looks like this is happening first, perhaps driven by the bacterium. We don't know, but it really looks like that because it just continues going up, and at some point these people become microbiologically positive. So as you know, many in this room have done a lot of work on, well, what happens to lymphocyte and monocytes in the blood? And in fact, for decades, people have been using the monocyte to lymphocyte ratio as a way to diagnose infection. And so we could look at this as well. And so here I'm just showing a few examples of genes. Of course, we can look at genes that we know are expressed by particular cells in the blood. And so I'm showing myeloid cells over here. This is a granulocyte gene, and you can see it goes up. Monocyte gene goes up over here. And then we have a T cell gene over here, which goes down, and a B cell gene, which goes down. And so we know now that when people have this type of inflammation, myeloid cells are enriched in the blood and lymphoid cells are depleted in the blood. And this kind of happens sort of all within 1 year of TB diagnosis. So it really seems to be secondary to this interferon response that happens really early on. And so if we summarize this again using modular analysis, so now many, many more genes than one gene, we really see this very, very beautifully. Granulocytes and monocytes go up, T cells and B cells go down. And in fact, we confirmed this by flow cytometry because we had PBMCs from these adolescents. And so you can see here are the monocytes going up and the T cells are going down. And so this is really a real signal, but it happens much later than we can detect with this interferon response, which really seems to be the thing that heralds progression. So I just wanted to make a point about the way I think this title has been interpreted by many people to suggest that perhaps the transcriptional signature that we see in disease is neutrophil driven. And I think what the authors meant to say was that the neutrophil signal was dominant, but perhaps not that it is the thing that's driving the signature. And I think our data supports that. Oops, last one before the conclusions. So, of course, one has to ask, well, we've got all this inflammation going on. We see T cells going down and lymphoid cells going up in the blood. What does this mean for the T cells that presumably protect, and possibly the B cells even, that protect us against TB, if we believe that? And so we looked at a few things. I've already shown you the HLA-DR result. We know that all the T cells in the blood actually become, or pretty much many of them, become activated and they express DR. And here you can see what happens and when that happens. But what's also interesting is that we see that both CD4 and CD8 T cell central memory cells go down. These are not antigen specific. This is the entire pool. And so we really need to look at the antigen-specific response here to see what's going on with those. But I just want to make the point that it's very likely that when people are in this progression state, and perhaps this is indicating subclinical TB, there may actually be real issues trying to vaccinate people during that time. And perhaps this is where therapeutic vaccination could be a very important intervention. So the take-home message from this is that progression from infection to disease can take months or even years. It's very heterogeneous, and what I showed you are modeled data. But when you look at individuals, some individuals go really fast within a couple of months, whereas others really do take a long time. It does look like the type 1-2 interferon response heralds progression to TB. That is really the first thing that we see. And the inflammation and cellular changes seem to follow. So there's kind of a sequential wave of things happening. Innate inflammation may be partly implicated in T cell activation, but we need more data to support that. And then we really see this phenotypic skewing that happens in progressors. And of course, this may have implications for vaccination that we need to think about and explore seriously. So with that, I'd like to thank many, many people, too many to name individually, some excellent collaborations with some amazing people, and of course, a brilliant team of people back in Cape Town, and many funders without whom this would not be possible. Thank you.
We have about 2 minutes left.
Thank you, Tom. Lovely talk, lovely data. How do you try and tease out direction of causality in some of this?
Yeah, I mean, we can't. It's impossible. We can guess. We can do experiments to see whether we can replicate something. So, for example, when we take whole blood and we add TB to it, we can see upregulation of the interferon response. When we add interferons, we can see upregulation of that. Of course, that doesn't mean that a virus isn't doing it in the humans. So, we need to do more work to try and figure that out. I think those are very important questions, and it may very well be that we should be looking at CMV infection or other viral infections in every single study we do from now on to be able to tell if that's really a major confounder.
So, Tom, thank you. Fantastic to see you put it all together. And at the very beginning, you said that we can't find a correlate of protection because we give BCG and we don't have a placebo control. I just wondered if you knew if anyone was thinking about the fact that we've had a global shortage of BCG vaccine up to 13 million doses short, and whether I'm kicking myself. I feel that as people who are interested in immune correlates in infants, we should have reacted to this more quickly and tried to see if there was an opportunity to actually do that and to collect those samples so that we can, out of that disaster, we can gain some knowledge on protective immunity against BCG. And is there still time, if people are doing this and if we could do it?
Yeah, it's a good point. It's a disastrous scenario. I don't know if anyone has pounced on this. And I think in most instances there are catch-up vaccinations happening, which of course have to take precedence. But I think it's a good point, and maybe it's even still possible. So we should really think about it and see if we can. It's going to be a difficult ethics application, I think, because if you are in a research environment and you can get BCG, then you should really give it.
Thinking of Ebola, things can happen really fastly.
Yeah.
But people think that the need is there, and I think we can make a strong argument.
Yeah, no, it's a good point.
Thanks, Tom. Beautiful talk. With the DR expression on bulk T cells predicting progression in 2 studies now, right? The vaccinated and in your gene expression signatures, could you see evidence of DR upregulation as a predictor?
Yeah. So several HLA molecules are actually upregulated as people progress. I think this could simply be that we see cellular changes in blood. So as you know, monocytes express a lot of class 2. So do innate cells, and so I think it's difficult to tease out really whether this is activation of T cells, for example, as opposed to changes in cellularity in the blood. But we do see these. We see a lot of, in fact, also DM, for example, HLA-DM and other molecules. So we really need to get into those data and explore them more.
So it's great that the gene expression correlates with the finding from the flow cytometry. So in parallel, the BCG-specific T cells, the frequency of those in the Nature Communications paper, that correlated with less risk of progression, right? So BCG. Have you been able to see any gene expression signature that in turn correlates with that?
Well, I think Helen is sitting on the data and has not presented it yet, so perhaps she can address that.
In terms of gene expression signatures in the same infant population where we saw the HLA-DR, then we saw BCG-specific T cells protecting. So we focused the analysis on the unstimulated PBMC, and LAG3 is the top immune activation gene that comes up along with HLA-DR. So there's definitely other T cell activation markers. And we do see differential expression between cases and controls. Definitely viral infection has a big impact on the transcriptomic signature there as well. And there are differences in the way that the CMV positive infants have got risk of disease compared to the CMV negative. But yeah, coming soon, yeah.
So thank you very much, Tom.